The whole needle dissolves, releasing the encapsulated drug. And then when you take the patch off, there's no sharps left. Finally, you could have a microneedle that looks more like a hypodermic needle, that is a hollow microneedle. And now you go from a dry but to a liquid formulation, and you make an injection, flowing the liquid through the hollow needles into the skin. There's some other variations as well, but most of the microneedle research that the community is doing falls into these general categories. I'm going to give a few case studies, few short stories on different technologies that leverage this. And the first one that I'd like to talk about is the dissolving microneedle and how we have studied that for vaccination. Second one, we'll go to the solid microneedle concept, where we've taken microneedles off of patches and put them onto particles that we call star particles, and we're using that for topical delivery in dermatology. Again with the solid microneedle, but giving it a very different functionality, we're turning these microneedles into electrodes to give electric pulses to the skin to cause a phenomenon called electroporation that we'll discuss more. Another approach, again using the solid microneedle, is using the microneedles in reverse. So we are making pores in the skin, but pulling fluid out of the body rather than putting drugs in. And I'll try to describe to you why accessing that interstitial fluid may be of interest. And then the final one goes to hollow microneedles, but now we leave the skin and we'll look at hollow microneedles for use in the eye. And I'll explain why that might be of interest as well. So with that preview, let's jump into the first story, which is the skin vaccination by a microneedle patch. Here is an example of some of these microneedle patches. And if we take a closer look at one of them, you can see an array of green spots. So those are microneedles that have been loaded up with some green dye to help you see them. There's a shiny area in the periphery. That's the adhesive to stick to the skin. There's a tab to grab onto that doesn't have adhesive on it. There are various designs. This is the general design that we like to use. And if you look closer still now, you can see these microneedles. You can see their dimensions. They measure hundreds of microns, so plenty long to get across that very thin stratum corneum barrier. The drug, in this case a green dye, is loaded selectively into the microneedle part of the device. You don't want the drug in the backing because that wouldn't be efficiently delivered. So we have a process to load selectively into the needles. And I can say a little more now about how we went about coming up with this design. And we considered a number of things. One of them was from a patient administration standpoint. So we want these patches to be very simple to use. We want minimally trained personnel. We don't want to have doctors and nurses have to do this, but people with minimal training and even patients themselves, when it's appropriate, would be able to self-administer these patches. There's no applicator involved. It's just your thumb. There's no reconstitution. That is, you have a dry formulation and the stability that a dry formulation allows. And you don't have to add water before injecting it because it reconstitutes in the fluids of your skin. The delivery is painless because the needles are so small. And likewise, there's no fear of the needle, which can be a barrier to people getting injections otherwise. From a manufacturing standpoint, we think this is a low cost and scalable fabrication method. And the method basically is this. We start out with an inverse mold in the shape of the microneedles. We then cast the first solution onto that mold, which contains the antigen or the vaccine or potentially a drug if it's not a vaccine. And that fills the needle cavities, forming the microneedles. We then cast a second solution on, and that has no antigen or active in it. And that creates the backing layer that holds everything together, but doesn't have a drug in it. Once everything is dried, we can pull it out and put it in a package. So the manufacturing process is essentially mixing up two solutions, casting them as two films and allowing them to dry and putting them in a package. There's a few more details in there to get it right, but I hope you can see that this is a process that is amenable to conventional kinds of pharmaceutical manufacturing environments and one that could be done at low cost. The expectation is that in mass production, millions per year levels of production, these would be less than $1 each. So competitive, say, with the cost of a pre-filled syringe. For transportation and storage, there's obviously a small package size. These microneedle patches are small, and part of the reason is we get rid of all the water. Most of what's injected in an injection is water with just a little bit of active that's in there. So we're able to make things very small. We have the improved thermostability of this dry formulation, and in often cases can get either partially or completely out of the cold chain. From a waste disposal standpoint, the needles dissolve. So there's no sharps waste when you're done. There's no possibility of reusing these patches, unlike used hypodermic needles. And there's a reduced disposal volume for whatever does remain to be disposed of. So that led us to use these patches in a variety of studies. And what I'd like to highlight for you is 2 clinical trials. And the first one is a clinical trial of influenza vaccination, which we carried out at our sort of sister university in Atlanta, at Emory University. There were 100 healthy adults in this study. We administered activated influenza vaccine provided by Seqirus. Seqirus is one of the major international flu vaccine manufacturers. And there are 4 study groups of equal size. We either had the positive control of intramuscular vaccination by nurse, then we had the 2 study groups of microneedle patch vaccination either by nurse or self-administered by the subjects in the study. They saw just a few PowerPoint slides of instruction, and that's all the guidance they received. And then finally, a microneedle patch placebo that was administered by nurse. So the results are shown here, the key results in terms of immunogenicity. The y-axis shows seroconversion. So that is the percentage of the subjects that had a 4-fold rise in their antibody titers. That's a standard measure of successful vaccination. And then you can see along the x-axis, there are the 3 strains of the flu vaccine. Today, it's a quadrivalent vaccine. At that time, a few years ago, it was a trivalent vaccine. So 2 A strains and 1 B strain in the flu vaccine. And then you see the groups of 4 bars. The first bar is the intramuscular vaccination, then the 2 microneedle patch groups, and then finally the placebo. For the 2 A strains, the seroconversion rate was quite strong. You can see 80% in that neighborhood for both intramuscular and microneedle patch vaccination groups. So all looking about the same and successful. When we look at the B strain, you can see that the intramuscular vaccination actually was not as strong. There's only about 1/3 of the subjects that seroconverted. And oftentimes the B strain is not as immunogenic, and that wasn't surprising. But what's of interest particularly is that if you look at the microneedle patch groups, they have a significantly higher seroconversion rate compared to the B strain. And that now relates to another feature of the microneedle patch that I didn't mention, and that is, well, obviously, it delivers to the skin. Vaccines are known to often be more effective when they are administered to the skin. It's been shown in various kinds of studies just by, say, intradermal injection, not just in a microneedle patch context. And that's because the skin is our interface with the outside world. It is expecting to encounter pathogens and to figure out what to do, how to mount a response against them. There are Langerhans cells in the epidermis. There's dermal dendritic cells. There's a lot of lymphatic drainage out of the skin. And these things all come together to make many vaccines more immunogenic in the skin. We didn't see that with the A strains because the response was already quite strong, but in the B strain, where there was room for improvement, you can see that the skin vaccination by microneedle patch gave us an immunological advantage, as well as the ease of access and logistical advantages that the patch can offer. From a safety standpoint, we can look at the site of vaccination on the skin. And what I'm showing here are some representative images. The application site was the wrist. So you're seeing a close-up of a wrist. And we did see mild reactogenicity at the site where the patch was administered. You can see there at day 0, perhaps you can make out there's a square in the center there. So that is where the microneedles have just gone into the skin, left there for 20 minutes. And then pulled away, and a little bit of mild erythema around it. That's from the adhesive stuck to the skin that just got pulled off. So, you pull a plaster off your skin, there's a very transient erythema that comes from that. When subjects came back 2 or 3 days later, you can see again some mild evidence, some mild erythema there at the site of patch application. By 8 to 10 days, again, very faint, but you can see something. And at the 28-day visit, you see nothing. So there was some notable erythema, again, mild, of no medical or safety concern, but perhaps of some small cosmetic concern. Overall, if we compare the microneedle patch to the intramuscular injection, the tenderness associated at the site where the vaccination was done was about the same with microneedle patch and intramuscular vaccination. Erythema and pruritus or itchiness, those are both greater in the microneedle patch group than in the intramuscular injection. Probably there was erythema at the site of injection, but of course you can't see in your muscle. You can see literally the immune response being mounted in your skin. And so we think that's what the erythema is. To support that interpretation, I'm not showing pictures, but if you look at skin after placebo patch administration, on day 0, it looks about the same. So there's just a little mechanical injury that's been done to the skin. But by day 2 or 3, or in fact, we even know from just other experience that we have, within a few hours, if you apply a placebo patch, you can't even tell where the patch was applied. So what you are literally seeing here at the days after patch application is the immune response taking place in the skin to the vaccine. Itchiness, there are itch receptors in the skin. There aren't itch receptors in the muscle, so that can explain why we perhaps had itchiness reported. And finally, from the pain perspective, the intramuscular vaccination had more pain than the microneedle patch. And that was both the pain of the injection as well as that sore arm residual pain that you can have after a vaccine has been administered. In terms of systemic safety signals, there was nothing notable there. You know, people have various things they report after a flu vaccine, and those were similar in the microneedle patch and the injection groups. So this work with flu led us to do a study more recently on measles and rubella vaccination. And we were motivated by the following, that measles has been a killer of children for a long time. In 2000, there were about 1 million kids who died of measles. And then there was an intensive measles vaccination campaign done. A lot of money was put into it, and that was lowered down to as low as 60,000 in 2020. It's crept up again a little bit, perhaps due to effects of COVID and people not getting vaccinated. But getting the vaccines out has made a remarkable difference and has saved a lot of lives. But we're not at 0. We're nowhere near 0. And it's not because of a poor vaccine. These vaccines are incredibly effective. A single dose will protect most people for life. Second dose, almost everybody. These are just kids who aren't getting vaccinated. They don't have access to the healthcare system to get the vaccines to them. So that's where we thought a microneedle patch could make a big difference. If we can have a patch that has some thermostability to it, so it can get out of the cold chain, and perhaps even more importantly, can be administered by people without advanced training, we can get patches out to these remote locations where they're needed and protect these kids. Measles is a similar kind of story in the intensive campaign to get, rubella is a similar story. In the campaign to get measles vaccines out, rubella vaccine was given as well. Cases have been brought way down, preventing congenital rubella syndrome. And that's good news, but we're still not at 0. And so the same motivation is there to get these vaccines out. So we carried out a clinical trial this time, not in Atlanta, but in the Gambia in West Africa. It was a phase 1/2 clinical trial. And this is for the American audiences to show where the Gambia is. This audience knows. Involved a number of collaborators. So the Medical Research Council of the UK has a unit that's been in the Gambia for many years where clinical trials are conducted. Biomedical is the company that made those patches. The Centers for Disease Control and Prevention, conveniently for us, located in Atlanta, but they were involved with the study and did all the serology. Gates Foundation supported it, and the vaccine came from Serum Institute of India. So the trial design here was a phase 1/2 trial. It was randomized, active controlled. It was double blind and double dummy. And so I'll explain what that means, which is every kid in the study got both a shot and a patch. If you don't do it that way, then you can't blind the study. Everyone knows who got the shot and who got the patch. You can't tell the difference. So every kid either got their vaccine by shot and a placebo patch, or they got a placebo injection and they got a vaccine patch. And so that way we could make it truly blinded. It was a age de-escalation design where we started out in some adults just to make sure that safety checked out. When that did, we then moved on to the toddlers, 15 to 18-month-olds. Those were kids who'd already gotten their first dose of measles vaccine and were now getting their second dose as part of the study. And finally, we moved to the infants who are getting their first dose. And these ages are commonly used in much of Africa, although I understand in South Africa, measles vaccination is now given at actually an early age, but much of the world is giving vaccines at these ages. We followed various things, so local and systemic adverse events, safety labs, unsolicited adverse events, and of course, the neutralizing antibody responses. And I'll just cut to the chase and show a little bit of data here. These data are from the infant group, so the most sensitive group, but similar things were seen in the older kids as well. And I'm showing now the seroprotection. So measles vaccine has been given long enough that if you have a certain antibody level, it's known that that correlates with protection against getting measles. The red bars are the microneedle patches. The blue bars are the subcutaneous injection vaccines. At baseline, there was just a little bit of background, some kids who'd been exposed to measles somehow and had some antibodies. Day 42 and day 180, you can see a very robust response where there's no significant difference between the response to microneedle patch and subcutaneous vaccination. The story with rubella is even clearer, where 100% seroprotection was seen in all of the subjects. So from an immunogenicity standpoint, things look promising. We also looked at safety and various things were assessed. I'm showing the reactogenicity here in the infants. So in terms of tenderness, we essentially didn't have any reports of tenderness after the vaccination. Erythema, there was some. So you can see in the upper left quadrant of the four, that is the microneedle patch site after the vaccine was given at that site. So a little bit of erythema is there, but also at the placebo site, there's a little bit of erythema. So just a mild skin damage that then can recover. Subcutaneous injection, none seen. What was notable was the induration or the swelling at the site, and you can see also some discoloration of the skin that could persist afterwards. That was specifically associated with the microneedle patch and the vaccine. Interestingly, you can see a delay. So it's not something that happens right upon patch application, but it happens a week or more later. And we think that is the immune response. So these are the infants. They've not been primed. They're naive to measles and rubella. And so the immune system is kicking in. It's a live virus vaccine, so it has some lifetime in the skin. And you're seeing then that immune response kicking in a week or so later, leading to some local induration. Interestingly, I'm not showing the data here, but when we did this in the toddlers, so they were not naive, they'd had a measles vaccination before, their induration response was immediate after the vaccine was given. So some interesting science, a minor cosmetic effect, no safety concerns were evident in the trial. So I actually have the safety data here, and I'm showing some of the key data. So these are at the application site, what we saw. The first points are the prevalence of the various indications, that the various adverse events that were observed. What's more interesting, I think, is when we subtracted them. So it was the prevalence in the group that got the microneedle patch vaccine minus the prevalence in the other group, which is the placebo microneedle patch, which is the same people as the subcutaneous vaccine. So there was one that was significant. You can see the very first bar, application site discoloration. So that was significantly greater when the vaccine was given by the patch. And you saw an example of that on the previous slide, what that discoloration looked like. When we look at the various other things, so upper respiratory tract infection, gastroenteritis, all these things, there was no significant difference. These are just, kids get sick sometimes. And so they sometimes did get sick, but it wasn't correlated with the vaccination by one route versus the other. So the safety profile, again, looked quite good. This work is proceeding on either later this year or early next year. We're expected to go to a phase 2B clinical trial that will take place again in the Gambia. Meanwhile, manufacturing infrastructure is being put in place. a pilot facility that can make 10 million patches per year is being designed and built. And that will then ultimately lead to enabling a phase 3 clinical trial that we hope will happen a year or two after the phase 2B, and hopefully clinical introduction so that we can reach those kids who are not getting vaccinated. So that was story number 1. The next stories are each a little bit shorter than this first story. We are motivated here to think not about vaccination, but to think about dermatology. That many drugs would have an advantage for being delivered right to the skin, right at the site where they are needed, but the skin is a really good barrier. So when you rub some dermatological ointment or cream onto the skin, the absorption is often pretty bad. And especially if it's a very water soluble drug or a high molecular weight drug, they're not absorbed well to the skin. And so you would either, well, you could give a local injection or you could take the drug systemically. And systemically means that you risk having side effects. And if you could deliver it locally right where you need it, you could get a high dose where you want it and minimize the systemic side effects. So based on that, we thought about using microneedle patches for that kind of delivery. But oftentimes in dermatology, we don't just want to treat a square centimeter, which is fine for vaccination, but you want to treat large areas of the skin. So that led us to the idea of taking the microneedles off of patches and putting them onto particles that could be rubbed on the skin. So here are some of these particles. They're 3 arms or 3 microneedles. It's kind of the Mercedes-Benz design, where you now take these particles, you add them into some kind of a topical formulation, and the rubbing action that you normally would do to apply something to your skin, that rubbing action causes these particles to roll and slide around and poke little holes into the skin so that the drug can be absorbed more effectively. These particles are made out of titanium dioxide, which is a commonly used whitening agent in various topical formulations, whether cosmetic or pharmaceutical. It's used in the white paint on the wall as well. So a very widely used and understood material. So here in the vial are some of these star particles. Basically looks like some white powder unless you look very carefully at it. You can see some of them on a fingertip. You can see them integrated into a cream, and well, you can't see anything. They just disappear into it. When we rub them onto the skin, though, they have an effect. So this is pig skin ex vivo. You're looking at a cross section through the skin where we either on the left-hand side just applied a dye, sulfurrhodamine, onto the skin surface, or on the right side, we pre-treated the skin, we rubbed the skin with some star particles, and then we put the dye on, everything else then being the same other than rubbing the star particles. At 1, 6, and 24 hours on the left, you don't see much. The drug or the model compound has not penetrated the skin significantly. When we have rubbed star particles on now, that molecule is penetrating very nicely and deeply into the skin. We did the same experiment, except this time with a larger molecule, a 4,000 molecular weight dextran with a fluorescent tag on it, and the story is quite similar. We're able to get nice penetration into the skin by rubbing star particles on. So we moved from fluorescent molecules to drugs that might be of interest and looked at 5-fluorouracil. So that's a drug that can be used to treat skin cancer. Methotrexate, that's a drug that can be used to treat psoriasis, typically given systemically because it has to be. We could perhaps give it locally and avoid the significant side effects that are often associated with systemic methotrexate. And bleomycin, a drug that's used to treat warts, it's given by injection at the wart, so we could perhaps avoid giving injections. And you can see the white bars are just a placebo, so rubbing some gel on the skin and then giving the drug, or rubbing the star particles on the skin and then giving the drug in the gray. And you can see an order of magnitude or more increases in the skin's permeability, that is the drug uptake into the skin associated with star particle treatment before applying the drug. We went a little bit further with the 5-fluorouracil, went with a melanoma model in the mouse and had 4 groups there where we either just rub gel on the skin, we just rub star particles on the skin, we just put drug on the skin. Those were our controls, or we gave the combination of the star particles and the 5-fluorouracil. And perhaps you can see visually in the first 3 groups, there are large tumors, and in the last group, there isn't. If we quantify the data, this is the tumor volume on day 12. Again, in those first 3 control groups, there wasn't much of an effect in terms of tumor volume, whereas in the group that received the star particles, as well as the drug, enabling that drug to penetrate more effectively, we were able to control that tumor growth. Finally, we've rubbed star particles onto humans. So here's the arm of a human subject. The arrow is pointing to the place where the star particles have just been rubbed on. There's no drug here. It's just star particles, just looking at safety and acceptability. And while you presumably don't see much there, and if you come back a day later, you still don't see much. So these star particles are very well tolerated, rubbed on the skin. They're described as a kind of a tingling sensation, but not something unpleasant, not something painful. And so we think that this bodes well for our ability to use star particles in a clinical setting. Story number 3. So now we're going to take the microneedles and do something quite different with them. That is, namely, to use them as microelectrodes to give electric pulses to the skin. And as the heading here says, it's piezoelectric microneedle electroporator. So I'll explain the piezoelectric part and electroporation. And our goal is for DNA and mRNA vaccination. So if we think about how DNA and RNA are administered, the best known method worldwide these days is the lipid nanoparticle. Pretty much everyone has heard of it, not just scientists, as a way of getting mRNA to be effectively delivered. But lipid nanoparticles really have some limitations. The lipids are expensive. The microfluidic-based manufacturing is expensive and complicated to do. And the side effects associated with the COVID vaccines are at least partially believed to be associated with the lipid nanoparticles, not with the vaccine itself. Also, that cold chain storage, which can require up to minus 80 degrees Celsius, is in part associated with keeping the lipid nanoparticles stable. So it'd be nice if we didn't have to use lipid nanoparticles. If we think about DNA and how it's administered, viral vectors are probably the most effective way to administer deliver DNA, whether it's vaccine or otherwise, but that's also complicated to work with. So we turn to a method that's been around a long time, namely electroporation. You can go back all the way to the 1980s and the early days of genetic engineering and find people using electroporation at that time as the dominant method for putting, then it was DNA, into cells. And today, I don't know, probably some of you in this room have electroporated cells before and use it as a laboratory technique. So these electric pulses will destabilize cell membranes and then allow genetic material and other things to enter into the cells, and the membrane then reseals spontaneously afterwards. So it's a nice method, except for the part where if you want to try to use it clinically, you're going to have this box that costs $10,000 and is really sophisticated, and you have to know how to use it. And then you apply the electrodes to the skin and you give these pulses, which can be hundreds of volts, milliseconds long, and the arm twitches and the patient is not very happy about it because you're stimulating nerves. So it's a good idea, but it has some real limitations outside of the laboratory. Well, that led us to thinking about how could we do it better? How could we get rid of this cost and complexity of the big electroporator? And how could we make it so that people's arms don't twitch and feel pain? And that led us to the idea for the electric pulse and having an inexpensive source of electric pulses to a barbecue lighter. So if you think about how a lighter works, there's of course a gas supply in there, but you need a spark to get it going. So you push a button or roll your thumb and you get a spark to come out. And what's going on is there's a little piezoelectric element inside of your lighter, and a hammer hits it. And piezoelectricity is the interconversion between mechanical energy and electrical energy. So the hammer hits the piezoelectric crystal, that generates a voltage pulse. And in the case of the lighter, you put the voltage across an air gap and you get a spark. So the spark lights your flame. We don't want a spark, but we want that voltage. So here's an incredibly inexpensive, widely available device that'll make a short, high voltage pulse. And that's exactly what we need for electroporation. So we went and looked into it, went onto Alibaba in China. They're making these lighters by the billions, and you can buy them. You can see here for 30 or 40 cents. And in fact, we don't need the whole lighter. We just need the little piezoelectric element, and that is almost free. So I think we have now addressed the issue of how do we get a high voltage pulse at low cost. We've gone from $10,000 to about a penny. So $0.1. So that I think bodes well for our ability to give low-cost electroporation pulses. We then incorporated into a device that involves, of course, the microneedles have to come back into the story, and the microneedles are the electrodes. So the reason why that's of interest is we don't want to stimulate nerves deep in the skin and cause twitching and pain. We want to keep the electric field just superficial, just in the epidermis of the skin. The epidermis is where all the cells are. The dermis has cells, but the epidermis is full of cells. So by having microneedles that just go into the superficial skin, the electric pulse only goes to the epidermis and avoids all those nerves that are deeper down. So here's the basic design. Everything above that red piece in the picture, blue piece in the schematic, everything above that, we just literally buy from Alibaba. That's the lighter element. This actually doesn't come from the barbecue lighter, but comes from one of those gas heaters that you may sit under on a winter's day at a restaurant. It's a little bit bigger, more robust piezoelectric element. In any case, we take that, we connect it to an array of the microneedles, and we have our complete system. So it's ultra low cost and simple to use. Just push a button. So it's because it's got the piezoelectric voltage source, less than $1 for the whole system. It's handheld, thumb operated, no battery, no power supply. It's targeted to the epidermis by using the microneedle electrode array, and thereby can avoid the nerve stimulation. And it doesn't involve lipids, it doesn't involve viruses, and that could give some advantages. So here's what happens when you use it. We can either take clamp electrodes, so these are larger electrodes that you clamp onto the skin with a conventional big box pulsar, or we can take what I just showed you, the microneedle electrodes with the piezoelectric pulsar. We then gave DNA that encodes for green fluorescent protein, and we're looking either NFOS, so on top, looking down at the skin, or below is a cross section through the skin. And you can see in both cases, we did transfect cells. You see the GFP being expressed, and you can then see on the cross section just a thin band of fluorescence, which is in the epidermis. So we've targeted the epidermal cells of the skin. We then moved on to doing a vaccination study. This was with a SARS-CoV-2 DNA vaccine, where the study design is shown as a prime and a boost a month later. Let me go to the key data. So these are the antibody responses. The first two bars are after intramuscular vaccination of the vaccine at either 10 or 100 micrograms, and you can see a dose response. We also gave intradermal injection of the vaccine, which looked similar to the intramuscular at the same dose. We then gave that lower dose of 10 micrograms with the electroporation that I just showed you, and you can see the immune response bumped up, that we got as strong a response from 10 micrograms as with electroporation as we did with 100 micrograms without. This work now continues, more work with DNA, and now we've also moved into the mRNA vaccination and are starting to get results on that as well. Story number 4 moves to microneedles in, again, a different context. And now we're not doing drug delivery. Now we want to access the interstitial fluid that exists in your skin. And I'll try to describe why that might be interesting. If you think about the biomarkers that we measure in medicine, they mostly come from blood. Occasionally saliva or urine, but blood is really the workhorse for getting biomarkers. But blood is only 8% of the fluid in your body. And there's all kinds of other fluid. About half of it is intracellular fluid. That's pretty hard to get. You probably got to kill the cell if you're going to try to access that fluid. But almost a quarter of the fluid in the body is the extracellular or interstitial fluid. So this is the fluid that bathes the cells and the tissues of our body. And it has information related to the blood because its source is the blood. But the blood goes through the capillaries and fluid leaks out of the capillaries, carrying with it oxygen and nutrients, comes to the cells in your tissues. Well, that's the interstitial fluid. The cells then metabolize, they give their CO2, they will release other waste products. That goes into the fluid as well, and then it can go into the lymphatic system, return back into the vascular supply. So there's a fluid there that's interesting, but we know actually very little about it because it's incredibly hard to get access to that fluid. So that led us to thinking about how we could get access. And of course, the microneedle is the example. If you poke your skin with something sharp, like a lancet or a needle, and you see what comes out, it's going to be blood that comes out. Well, we don't want the blood. We want the interstitial fluid. Well, if you do something very gentle on the skin and it doesn't bleed, nothing comes out. So we have to kind of coax it out. So we use these microneedles. They're small. They're just 200 microns long. They just penetrate in, trying to avoid hitting blood vessels, but nonetheless making punctures across the stratum corneum. And then we apply a mild suction to pull the fluid out. And when we do that, we're able to get these droplets of fluid onto the surface of the skin. They're a little hard to see because they are clear. There's no blood in them. It's just a clear fluid, but we can collect microliter quantities of the interstitial fluid by sucking it out through these little microneedle pores. The process takes about 20 minutes to pull the fluid out. So we did this. We did a small study in 21 healthy adults, where we collected actually 3 different fluids. We collected the interstitial fluid, as I described. We also had a kind of a control suction blister fluid. If you apply heat and suction to the skin for about an hour, you can get a blister to be formed, like, blisters that you can get normally in life. You pop the blister, you pull the fluid out, and that's related somehow to this interstitial fluid, although there's more injury that's been done to the skin to collect it. And then we collected venipuncture plasma and compared the biomarkers that we found. So this was an analysis of metabolites, of small molecules by mass spectrometry. There were about 10,000 compounds that we detected, and we created this Venn diagram. You can see 69% of the compounds that we detected were common to the interstitial fluid and the plasma. And so that's perhaps not so surprising. As I'd mentioned, the source of the interstitial fluid is a leakage out of the capillaries from the plasma. So a lot of biomarkers can move between the 2 compartments. It might be of interest to measure a biomarker that you could otherwise find in blood, but can also find in interstitial fluid, because interstitial fluid doesn't clot. So if you want to make repeated or continuous measurements, that's pretty convenient. And in fact, the one thing that is routinely measured in the interstitial fluid is glucose. You may know about these continuous glucose monitors that diabetic patients can wear. So these are sensors that are embedded in the subcutaneous space, and they stay there for a number of days, continuously measuring glucose concentration in the interstitial fluid of the subcutaneous space, which is in communication with the vasculature. There's about a 5 or 10 minute delay between a glucose concentration change in the blood, and then you see it in the interstitial space by these glucose sensors. We can then see that 11% of the markers that we found, they were unique to the plasma and not found in the interstitial fluid. So if those are ones you want to measure, those are not the ones to look for in interstitial fluid. So it doesn't have everything, but it has a lot. But I think particularly interesting is 17% of the compounds that were detected were unique to interstitial fluid and were not found in the plasma. And this reflects, for example, these metabolic processes taking place in the skin, and we are seeing biomarkers associated with that. There can also be things that are absorbed via your skin, and those are localized preferentially in the skin, so you might come up with them as well. We did an additional study trying to look at the dynamics of what's happening in the skin versus in the interstitial fluid. And we did a kind of pharmacokinetic study with the favorite drug of Atlanta. Atlanta, as you may know, is the home of Coca-Cola. So caffeine is the drug of choice in Atlanta, and we gave people Cokes to drink, either caffeine-free Coke as a control or a regular diet Coke. And we followed the caffeine concentration in the bloodstream over time, as well as in the interstitial fluid. The black data points show the subjects who drank Coke with caffeine, and you can see the interstitial fluid and the plasma track each other well. The white data points, those are for the caffeine-free, and so that's some background signal. We calculated the pharmacokinetic parameters associated with these curves, and the point isn't to show you the detailed numbers, but rather we then did a statistical analysis, and there's no significant difference in any of the pharmacokinetic parameters, whether the caffeine was measured in the plasma or in the interstitial fluid. So that suggests interstitial fluid is something that can be accessed. There's information in there of interest. And our next step in this is to now access this fluid from more people and learn more about what is in the fluid and what it could offer in medicine. The last story I'll tell moves to the hollow microneedle and in fact moves out of the skin and into the eye. I should emphasize here that I've advocated for the microneedle as something that is painless and people can self-administer. That argument disappears now. That is not the argument for ocular delivery. The argument here is targeting, is the ability to give an injection to a part of the eye that would otherwise be hard to access, but done by a doctor in a clinical setting, not for home use. So here's a cross-section of the eye. The cornea is shown at the top and the optic nerve at the bottom. The lens is in the middle. And the example that I'd like to give is for steroid delivery. So steroids are often given to the eye to treat inflammation. And in the back of the eye, macular edema, so swelling in the macula, the most sensitive part of the retina, can be treated by giving an injection into the vitreous cavities. Needles insert in the eye. That vitreous cavity is filled with the steroid drug. Some of that drug goes where you want it to go, so to the site of action in the retina. But some of it gets into the front of the eye as well, and it can cause glaucoma and cataract. So you would really rather the drug didn't go throughout the eye, but if it could be more targeted, that would be beneficial. So if we can get the drug just to the site of action and not other parts of the eye, we could reduce those side effects. That led us to the idea of using a hollow microneedle to target the suprachoroidal space. The very outer layer of the eye is the sclera. That's the literally white colored part of the eye. Interior to that is the choroid. That is a vascular tissue. Interior to that is the retina, the nervous tissue, and finally the vitreous humor. It's basically an inert hydrogel that fills the interior of the eye. If you make an injection at the interface of sclera and choroid, there's a potential space that exists there. So the path of least resistance for the fluid is not to go across these dense tissues, but to go between them and fill the gap between the choroid and the sclera. That's called the suprachoroidal space. And now you can see how we can get an injection to follow just the sclera and the adjacent retina and not flood the whole eye. Here is the microneedle that we have used, shown next to a conventional hypodermic needle that's used for intravitreal injection. Closer view shows you it looks a lot like a hypodermic needle. It's made of stainless steel. It's just a lot smaller. The whole needle is smaller than the bevel portion of a conventional needle. This one is a 0.5 millimeter long. We use those in rabbits. In humans, we get to just under 1 millimeter in length. And if you make an injection into the rabbit eye, you can see this. So this is a cross section through the eye. The cornea is the clear part shown on the lower right, and on the back would be where the optic nerve is. You can see the thin band of fluid. It's in that suprachoroidal space where we have made the injection. You can see a little leakage onto the surface in this case as well. And that has ultimately led to, through multiple clinical trials and various other efforts, to a product. It's called Xipere, delivers triamcinolone acetonide, so a long-acting steroid that dissolves very slowly over the course of about a month, so you don't have to get these injections every day. You get them very infrequently, and it is approved for treatment of macular edema. And so we hope that there can be other things that use this injection modality, namely there are phase 2 clinical trials going on right now for a treatment of macular degeneration, which is one of the leading causes of blindness. So that brings me to the end. Hopefully you have some better understanding about what microneedles are and all the various things they can do, whether we use them as a simple to administer patch for vaccination, can rub them on the skin for dermatologicals, we can turn them into electrodes for electroporation, we can use them in reverse to pull fluid out of the skin, and we can use them for targeted injections in the eye. There are many people who have contributed to the work. This is the research group as it looks today. And I'd like to acknowledge the various funding sources that have supported the work and be happy to answer questions with whatever time that remains. Thanks.
Thank you, Mark, for sharing your fascinating research. And I think probably, all of us have been most impressed in terms of how you can take one area of research and have so many diverse applications. And I think once again, you've highlighted to all of us that if we want to transform the treatment of various diseases, it is not only the API that is important, but it's rather how do we develop innovative delivery technologies. And I think that's what we take most from your presentation. So I'm sure there's many questions from the audience. We have time for a few. Is there anyone that would like to go first? Or maybe whilst you're thinking, maybe I can just ask. With the star particles, you showed us the in vivo efficacy studies. So I think what my colleagues and I would be interested in is a lot of us go up to in vivo efficacy, with our mice models, etc. But what else would we need to do in vivo to actually show the potential of our products to, for example, a pharmaceutical company that would be interested in investing in our technology? From a university point of view, what else do we need to do in vivo? Because we are at the efficacy point.
Well, of course, the hallmarks for any medical product is safety and efficacy. And so I think that the degree to which you can show efficacy in as relevant a model as possible is going to carry weight. So, I mean, mice are often good models, and that's where typically we start with things. If you have the opportunity to go to a larger animal model, I think that carries more weight. There's also a question of just how good the model is, and that depends completely on what the indication is. And some models are accepted as very relevant to what happens in humans, and others are more distant. And so that would be a judgment call to make. From a safety perspective, that gets a little more complicated, I think, because when you're looking for efficacy, you're looking for one thing to go right. When you're looking for safety, you're looking for a million things that could go wrong, right? And so kind of doing a full safety workup can often be really expensive to do. So I think it's difficult for universities to do a proper, full tox study and a full study like that. But if you have the opportunity to do it, I think that will carry a lot of weight. Finally, anything you can do in humans is beneficial. So we have the advantage that using just say, pressing microneedles into the skin or rubbing star particles on the skin without a drug, that already can answer some significant questions about tolerability and safety. And that's something that you can readily do in humans. You need ethics approval for it, but you don't have to go through regulatory agencies. It's not like the much more cumbersome process if you want to administer a drug to a human. So the degree to which a placebo formulation has meaning, you might try to move into humans with that sooner, because that is something that academics can often do.
Thank you, Mark. So I'm sure there's some questions from the audience. We have Prof. Mashige.
Information has side effects, particularly as you mentioned, cataracts and glaucoma. Have you tried some of these microneedles for the treatment of common infections like uveitis? I know you've mentioned the macular edema, but most of the common infections that we encounter, particularly in Africa, is your uveitis, the anterior segment disorders. Have you tried it for such treatment?
So the short answer is no. When I think about microneedles for the eye, one option is what I described, namely the suprachoroidal injection, and it's got the nice targeting to the choroid and the adjacent retina. A limitation is that it requires some significant expertise to administer it. So I think it makes most sense to use that in combination with a long-acting formulation. And triamcinolone acetonide is a nice example of that. But if you have to give it every day or every week, I mean, forget it. So that is going to be a characteristic. There is the possibility, which I didn't talk about, of using the microneedle to help get a drug into the front of the eye. So, with eye drops that we take and they're absorbed, the drug is absorbed across the cornea, but many drugs are not absorbed well across the cornea. And the argument goes the same as the skin argument as well. There's an epithelial barrier layer there. We have done work with the microneedles to get across the cornea as well. And one example is we've created a drug depot within the corneal stroma. So the center part of the cornea is avascular. That's why we can see. So there are blood vessels blocking our vision. And as a result, clearance is very slow out of the corneal stroma. So you can also have something that can be long acting within the corneal stroma. And that's another angle that could be useful.
We have time for at least one more question. Go ahead.
What do I do before I use
Sorry, could you speak?
I can't hear you very well.
what do I do before I use a microneedle on my skin, like the preparation for it? So if I need a microneedle, do I need some ointments before I use a microneedle, or do I just put it on my thingy on my skin?
Yeah, so the question, if everyone couldn't hear, was about how do you prep the skin before applying a microneedle patch. And what we do is we do an alcohol wipe. So wipe the skin with alcohol, let it dry, and then we put the patch on. Quite frankly, it's not clear if that's necessarily needed. In the United States, standard of care is if you give an injection, a hypodermic injection, you do wipe the skin. WHO recommendations are before an injection not to wipe the skin because there's no evidence that it's needed. In any case, we've wiped the skin before the microneedle patches, but it is possible. I mean, if the skin's dirty or something, you certainly want to be clean, but it's not clear that you necessarily do need the alcohol wipe.
So in closing, Mark, I'd like to thank you very much for visiting us at UKZN, for interacting with our staff and students. And I think most importantly, for inspiring us to reach global excellence in research and to start thinking about how we can actually commercialize our research. So thank you very much. We have a little gift. Maybe we'll just allow one question from our student.
I don't remember him mentioning the effect the microneedles have on our eye before and after they poke.
Yeah, so the questions about microneedles being administered to the eye and safety questions. So the way that the microneedle is administered for those injections is essentially identical to how intravitreal injections are done. So in now common clinical practice, the longer needles are pressed just back from the limbus. So the limbus is where the clear cornea, which then has the color of the eye, the brown or other color, to then the white part of the eye. So just a little bit back from where the white starts, you stick the needle in and you make the injection. So the suprachoroidal is done exactly the same way, but just a much smaller needle inserted. So the safety profile is similar to a conventional intravitreal injection, although at least a theoretical lower risk of endophthalmitis, so lower risk of an infection getting into the eye, because the needle doesn't go all the way in, it only goes partly in. So there is some pain associated with that injection. So you usually give an eye drop to locally numb the site. Then you give the injection. The eye can be a little bit sore afterwards. And again, the experience is very similar for the patient to an intravitreal injection.
Thank you, Mark. Thank you.
Thank you.
Oh. What? Oh, okay.
All right.
Okay.
Okay.
There's a
When I got turned out,
And finally, I'd like to thank Pamela Adams and her team at Public Relations, leadership and staff in the research office and the School of Health Sciences, and also my precious team in the Drug Delivery Research Unit for hosting Professor Prausnitz. And thank you to all of you and our online guests for attending this presentation and for your participation. Thank you.